This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 453, recorded on July 31st, 2017. This episode of TWiV is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with your first purchase and free shipping by going to blueapron.com/TWiV. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. We are coming to you today from the Rocky Mountain Laboratory. That's in Hamilton, Montana. And we are at a meeting called the Vector-Borne Viruses Symposium. And I was asked to come here and record an episode of TWiV. And I know that my colleague Dickson likes it here very much. So I invited him to join me. So welcome, Dickson.
Yes, thank you, Vincent, for footing the bill to get me out here, because I would have come anyway.
Well, you didn't tell me that.
I wouldn't have come to this meeting, though. You would find me on the river fishing.
So that's why Dickson came, because he's a fisherman. He's been here many times. Yeah. And the science is not enough to bring you anymore.
No, I would have come.
Well, thanks for coming. And we have picked from the speakers at this meeting a very special guest, someone I've wanted to have on TWiV for a very long time, has been in virology a long time with a wonderful, productive career we're going to hear about today. She is a professor at the Johns Hopkins Bloomberg School of Public Health. Diane Griffin, welcome.
Thank you.
To the show.
All right.
Wow, you have a lot of fans.
Oh, yeah.
This is terrific.
Stack the audience.
All right, so we have about an hour. Hey, could someone close that door back there for me, please, so I don't hear the crowds
Cheering in the back.
cheering outside? Thank you very much. So what we're going to do is go through a little bit of Diane's history, and then I've gone through some of her papers, and we're going to touch on some of them and talk about them and what they mean. And then at the end, as I told her, we have some philosophical questions to ask, which you may or may not have answers to, but that's fine. So let's start with the very beginning. Where are you from originally?
Originally, I was born in Iowa City, Iowa, but I only lived there for 6 weeks. My father he was a graduate student in geology and just completing his PhD, and he went to work for the oil business, and we moved to Oklahoma City. So I grew up in Oklahoma.
Mm-hmm. And you went to high school, through high school?
High school, all the way through high school in Oklahoma.
And where'd you go to college?
Augustana College in Rock Island, Illinois. So the Midwest.
Still Midwest, okay.
Yeah, but right.
What did you major in?
I was a biology major. It was the days before you didn't have all these specialized majors. You were chemistry or biology. Yes.
But was there some, why did you do science? Was there some family influence? Was it a teacher?
Well, as I say, my father was a geologist, so there definitely was some science family influence, and he had done his PhD thesis on the Teton Mountains, north end of the Teton Mountains, the geology of that. So family vacations were very oriented toward mountains, toward education. Road cuts were very important to look at when you travel with a geologist. My mother was a teacher. She taught preschool, actually. And I just was interested in science, certainly in high school. Realized that was a major interest. And in college, took a course in microbiology, and so that sort of started tending me in that direction.
Right, okay. So when you took family trips, you collected rocks?
We did, well, we were actually discouraged from doing much collection, because you leave things as they are.
That's right,
but we certainly looked at a lot of rocks and certainly plants, etc. A lot of hiking.
So you majored in biology. You took a microbiology course.
I took one microbiology.
And then, so obviously you went on in science. So was there some point in college that you decided you wanted to go further in science?
Well, I think I was a little undifferentiated and didn't really know
I didn't really,
well, I knew I was interested in science, that's all I could tell you, and sort of what were the next logical things to do for somebody who was interested in microbiology. And so at least at this particular time and at this particular college, going to medical school was not considered a very viable option for most women. And so doing a PhD, actually in microbiology, it was a default option, basically.
That's interesting.
Yeah.
Because Joan Stites, who we interviewed 2 years ago, she said she was going to go to medical school, and she saw all the doctors were men, so she didn't want to go. She decided to get a PhD.
Yeah, well, but then I had a sort of a devious path because I went as a PhD student, and then I transferred into the medical school and ended up doing an MD PhD because I was really most interested in the disease processes of
So this was where at
So I went to Stanford
for your PhD initially,
for a PhD.
right? What department
initially.
was that in?
The microbiology department.
And who was the chair at the time?
Sidney Raffel.
Wow.
Oh,
know
right.
him.
Oh,
He
sure.
was sort of a pioneer in cellular immunology. He studied TB primarily, but the delayed type hypersensitivity reaction was a specialty of his.
And then you were in that program, and you picked a lab already to do research in before you went into the MD program?
Right. I switched after I went into the medical school the next year. So I was there for a year as a straight PhD student.
Yeah.
But I pretty quickly recognized that I was really interested in the disease processes, and so I was totally naive. I applied only to Stanford because I was there. I thought that was a reasonable option.
Not a bad choice.
No, it wasn't, but I was kind of naive to think I was going to get in. But anyway, I had this one issue, which is once I decided that it turns out there were some requirements for medical school that were not requirements for graduate school. I'd done the GREs and that sort of thing, but I'd never taken the MCATs.
Oh, yes.
Turned out you had to take the MCATs before you could apply.
Right, that's right.
And organic chemistry.
And of course, and of course, they were being given for the last time next Saturday. So it turned out that if you got your money there, they would allow you to take it, even though you were supposed to have applied many weeks ahead of time.
So what did you do your thesis work in at Stanford?
It was an immunology project that was mainly, at that time, immunology, actually, even though Raffel was kind of a pioneer in cellular immunology, they really had not defined the various lymphocytes and that sort of thing. So immunoglobulins were really the thing at that point, and it was the early days of molecular biology with respect to immunoglobulins. So I worked on an antibody project.
Okay, and then what was your next step in your career? Did you do a postdoc?
Well, then I went to medical school.
She did a residency.
So you got your PhD first.
No, I ended up a gerrymandered MD PhD program. At that point, at Stanford, it was a wonderful place for me to be because Stanford had a 5-year MD program, and it was stretching the first 2 years into 3 so that you had half days in all summer to do research. So I pieced those together.
So in total, how many years you were there?
It was 6 years.
Six-year
It
program.
ended up being a similar kind
And
of.
then did you do a fellowship after your MD?
I did 2 years of clinical training at Stanford in internal medicine,
Okay.
and then moved to Johns Hopkins, and that's where I did the real research training, and besides what I'd done for my PhD, but that's where the fellowship, that's where I really got started in virology.
So you went to Hopkins as a research fellow.
I went to Hopkins as, again, kind of a combined fellow, but it was research, and also I did some clinical infectious diseases at the same time. But the formal thing was a postdoctoral fellowship, really.
Who was that with?
Dick Johnson.
Okay. Who just passed away last year, right?
Yeah, right, yeah.
And you wrote a nice obituary for him, I remember.
He was an important mentor.
Yeah, and you said in this nice obituary that he coined the phrase neurovirology,
Yeah,
right?
he did, right. and he was a real pioneer in pathogenesis-oriented research. So
Now, I met him several times. In fact, I had his book.
It was a good book on.
What's the name of it?
I think it's maybe Neurovirology, something like
about
that.
viruses in the nervous system.
Right.
And when I went to Columbia, it was like my Bible. And when he died last year, I took it out, and it was all underlined and notes written in the margins. He was really very good.
And he was a good writer.
And a nice guy as well.
Yeah.
And so he trained you. You used to train Janice Clemens, is that
Janice
right?
Clemens, Bill Narayan, Henry McFarland. There were a lot of us in the lab at that time.
No, he certainly influenced my thinking about viruses in the CNS.
Right.
And probably you as well. Right?
Yeah, completely.
So then after that, you stayed on as a faculty member at Hopkins, right?
Yeah, so,
And
right.
you've been there ever since.
Exactly.
Okay.
Did you ever use your MD to practice medicine?
Oh yeah, no. So my appointment at Hopkins was I had a joint appointment in medicine and neurology, but I'm not trained as a neurologist, and so I did attending on the infection. I was never very good at it, I have to confess. But I always attended. Anybody who's a clinician here will understand this. I always attended in June because that's when the fellows were totally trained and could do everything.
So if you're on an airplane and someone calls out, is there a doctor on the plane? Would you
I
respond?
always hide. Hope that no one's noticed.
Right.
Okay. So you're now a complete research scientist.
Yes.
So I looked at PubMed, and your first paper, I think this is right, August 1972. Does that sound right?
Right, yeah, that was probably my PhD, so.
And is that,
I
that
don't
was on
know.
SynBis? It was on SynBis. That would have been at
No, there
Hopkins.
was at least one paper before that, but
Okay, so
not that
let's-
it was a prominent
Let's
paper.
say this is your first one. It's specificity of the inflammatory response in viral encephalitis, part 1, adoptive immunization of immunosuppressed mice infected with SynBis virus.
Right.
So was that your first SynBis?
Probably so.
So why?
I'd have to-
How did you start working on SynBis?
Well, so as I say, I went to work in Dick Johnson's lab. He was just newly recruited to the Department of Neurology there.
Okay.
And he had just come back from a fellowship maybe a year or two before where he had in Australia and brought back SynBis virus as an interesting model system. And so it was my assigned postdoc. I'm still working on my postdoc project. I assigned postdoc. Everybody in his lab had a different virus. You could do that in those days. Each fellow had their own virus to work on, and so mine was SynBis virus.
So this was a virus native to Australia?
Well, it is actually, it's extraordinarily widespread. It's the most widespread of all the alpha viruses, actually. And so, yes, it's in Australia, but it's in Africa, it's in Europe. It actually only causes clinical disease in South Africa and in Northern Europe, Sweden, Finland, etc.
And what is the host range? See, I'm the non-virologist in this.
Right. Well, it's a mosquito-borne alpha virus, and the reservoir are really birds.
And people get infected, right?
People get infected. Oh yeah, if you just look at the seroprevalence, it's extraordinarily widespread. It's just people don't get clinical disease. But it may be the next emerging, and maybe the next Zika.
Yes.
Because it hasn't been introduced into the Americas. So it doesn't exist here in the Americas. It's Asia, Europe, and Australia, Southeast Asia.
And what's the mosquito vector, do we know?
It's a Culex mosquito.
Culex mosquito? That's interesting that it's not here yet. Now, why did Dick Johnson bring it from Australia, you said, right?
Because he thought it was he was interested in studying viral encephalitis.
Yeah.
The disease caused encephalitis in mice, and he thought it would be a good model system, which it turns out it has been.
Yeah, you've worked on it since then, right?
Right.
Essentially. So it did infect mice, and that was part of the appeal that it caused neurological disease in mice.
There was two kinds of appeal. It caused neurologic disease in mice, and at a time that there was really very little understanding of viral encephalitis at all. So it was a model system. Plus it was safe. Basically,
Right.
it didn't cause very severe disease in humans. So.
Are you seropositive?
Oh,
Have you
yeah.
ever- Yeah? You've checked?
I checked a lot of mice.
Do you
Yes,
still-
I don't. Yeah.
Do you still work in the lab?
No.
How long ago did you stop?
Oh gosh, that's a good question. I don't know. It sort of was a gradual process. I used to come in on the weekends and do experiments, and now
Do you miss
I
it?
write grants and papers.
You miss doing experiments?
Oh yeah, I do. And actually, for I don't know if you're going to talk about measles at all, but for a long time we had measles projects in both Zambia and before that in Peru, and that was always my chance to work in the lab. I would go and work there for 2 or 3 weeks and didn't take any technical help.
Yeah, I stopped long ago, but I'm not sure I would be able to work. And the only thing I can do is a plaque assay.
But
Right.
you do them very well.
I do a lot of them. I used
All I can say is that once somebody wanted to take a picture of me in the lab for whatever reason, and so I was there and I said, well, I'll just take something out of the incubator that looks like. My postdoc came running in. He said, "That experiment
yeah,
is
That's right.
can't just
plaque
touch anything.
Or
Yeah.
it wasn't." Yeah.
All right, let's talk a little science. The next paper I noted, which I think kind of is a trend in your career, 1976. This is your first sole author paper. So did Dick let you publish on
did.
your own? Oh, okay.
Right.
It's the same with John Stites.
Yeah.
She worked with Jim Watson, and he let her publish on her own. It's really interesting. I don't think that exists anymore. People let
Right.
people do that. You can't survive unless you have papers, right? In 1976, role of the immune response in age-dependent resistance of mice to encephalitis caused by SINBIS. And that's something that you worked on for many years, this age dependency. So how did you get onto that? What's the story?
Well, it was thought at that time. Well, first of all, actually, Dick had made the observation that, in fact, the observation had been made when the virus was first isolated, that if you infected newborn mice, they all died. And if you infected 4-week-old mice, they all got infected, but they all survived. So this observation of age-dependent susceptibility had been made in the 50s. And then Dick had written a paper that showed that that the virus replicated much better in young mice than in older mice, so that it looked like there was a tissue, basically that there was a restriction that was age dependent in virus replication. And so that paper really tried to look at whether, but most people thought it was because of the immune response, that there was a difference in the immune response, and young animals couldn't respond and older animals could. And so that paper looked at that hypothesis, which it turns out there wasn't a difference in the immune response. It again just reiterated the fact, or emphasized the fact, that it was really a restriction that happened with cells when they matured. They now no longer were as susceptible, both with respect to virus replication as well as cell death, actually.
Okay.
I was just going to add, if I were listening and I didn't know any better, I would say, well, maybe there's a blood-brain barrier effect also. As you're young, the blood-brain barrier is open, and by the time you get to be an adult, it's closed. Or at least it's more difficult to transmit.
That's true, and this age dependence for young mice, it doesn't matter whether you're infecting them in the brain or peripherally, they get neurologic disease and die. Older animals, that's not true, at least with the strain that we're using. There are strains that are neuroinvasive, but to infect the nervous system, you have to either use intranasal or intracerebral inoculation to be sure of that. So there is a restriction that is at the blood-brain barrier somehow.
Okay.
I don't really know what.
That's paper number 10.
No,
I don't have a number, but I think we're going to return to this, right? Because I have some other subsequent papers.
All right.
I think so, but I could be wrong. All right, the next one that I noted. So Diane has over 300 papers. I didn't want to do all of them here.
We only have an hour, Vincent.
I know
We can
that.
go through
I'm
them
watching the
all.
clock, don't worry. Just some highlights. 1992, persistence of viral RNA in mouse brains after recovery from acute alpha virus encephalitis. And this is interesting in light of today's discussion, because now we know that other RNA viruses persist. But back then, well, that's a good question.
Right.
Was this surprising back then that there was persistence of an RNA virus?
Completely.
Especially one that it would cause an acute infection, right?
Completely. I absolutely did not anticipate that, did not know that? No. So that really started us getting interested in persistence and the fact that acute RNA virus infections are not necessarily so acute. Even though the disease manifestations are acute, the viruses stay around for a long period of time. And we heard about it in Zika already.
So if you infect female mice, do their offspring die because they cross the placenta and kill the newborns?
No, because the animals develop an immune response, a great neutralizing antibody response, a great T cell response, but it just takes a very long time to clear the viral RNA. Again, like was talked about today, we can't. We can't. I mean, I can't say we've tried everything, but recover infectious virus. But the genome is there, and the immune response is ongoing. So protein is still being made. Yes, that was I think one of the first descriptions that this
is 1992. It's a long time ago. 90s doesn't sound like a long time ago, but we're now in 2017.
It feels that way.
And you were looking at RNA probably by northern blots back then.
Yeah, I was trying to remember what the technique
was
that we would use. But
was there PCR?
No, I think it was. No, I think it
maybe
was
PCR.
Yeah.
Okay. And you found that you could not find infectious virus, but there were RNA fragments probably.
Right.
Which is now,
And they were. You could find multiple parts of the genome.
Right. So, and we don't understand why there are fragments, right? Why the virus is breaking up at all, and why it's persisting.
Right.
I mean, if it can't replicate, our old thinking of RNA is that, gosh, it's so fragile, it's not going to be around very long. But
right.
Apparently, in certain places, and this is in the brain.
But this is in the brain, and so that was what we thought at the time. Was this makes sense because the tropism of the virus in the brain is neurons, so it's really only infecting cells that have that are basically have a very long lifespan, and if the process of the infection itself or the immune response doesn't kill those neurons because the animals recover, then maybe one of the price, in fact, that was our interpretation, that the price you paid for being able to recover from an infection of your neurons was that your neurons never really became completely free of the virus if they were going to be allowed to survive, basically.
I think later you showed this happens in humans infected with measles as well.
Right, yeah.
Right?
No. So we assumed, as I say, it was because it was a long-lived cell that the host could not afford to do without, was the reason that this was happening. But then when we found it for measles, that's lymphocytes, monocytes, these are all cells that can be easily replaced. It was still the case that the virus is around for a long time.
So is that because those cells,
the RNA,
are not dying, or?
Well, they have to not be dying, or they presumably, or you wouldn't still have them around. So I can't say that we totally understand, but we think it's an important aspect of, quotes, acute RNA virus infections.
Is there any way to get them to replicate by inhibiting something in the host?
Well, so people often ask that about Synbus, just immunosuppress the mice.
Right.
Except that, as I'll talk about tomorrow a little bit, the antibody response is incredibly important for suppressing virus, for clearing virus from neurons. And the antibody response, first of all, the half-life of antibody is very long, and B cells that produce antibody also are very long-lived cells. So it's very hard to get rid of an antibody response by immunosuppressing an animal. So we've never really tried to do that. But
So we know now that Zika can persist, Ebola viruses can persist, and I wouldn't be surprised if people look, they'll find other
think
viruses.
if you looked at any, quotes, acute RNA virus, you would find this.
In fact, when I came to Columbia, a guy named Jim Miller, who's a neurologist, worked on polio. He got it to persistently infect mice for months.
Right. Because, again, that's a neuronal infection.
Yeah. So keep your eyes peeled. In coming years,
syndrome.
There's going to be more persistence of virus. But it's not infectious virus. That's the key. It's pieces of RNA probably, right?
Yeah. as I say, we've never been able to recover it, but with Zika now and Ebola, we know that in some individuals it is infectious and can be transmitted. So,
right. Okay, now in 1993, a year later, a very cool paper, conversion of lytic to persistent alpha virus infection by BCL2. Tell us about that one. How did you get to, all of a sudden BCL2 is involved in apoptosis, right?
Right, and that was it. We had observed that that actually Synbus virus. I was working with Marie Hardwick at that time, who knows a lot about apoptosis, and we had observed that actually the way the virus killed neurons, particularly in young mice, because then the neurons did, in addition to replicating a lot of virus, did die. That the process by which they were dying was an apoptotic process. So then the next logical step was to say, if you took an inhibitor of apoptosis, could you then get the virus to not kill the cells? And Synbus and so we did it a couple of different ways. We had cells that overexpressed BCL2, which was at that point one of the earliest known inhibitors of apoptosis, and we also expressed it in the virus itself, and both of those things would allow virus to continue to replicate, usually at a somewhat lower level, but the cells to survive.
So these are, in this paper, conversion to a persistent, when you put BCL2 in the virus, is that correct in that time?
We did it both ways, as I say. We both put it into the cell to overexpress, but we also put it into the virus, and then you could get long-term virus replication with cells that survived.
Is that that's in a cell culture, right?
In cell culture.
But not in animals.
Exactly. Not in animals.
Okay. And that gave you some idea about what was involved in persistence, maybe, right?
Right. And it was one of the things we kind of pursued with respect to age dependence also, whether what allowed mature neurons to survive was, and they do upregulate expression of a number of they become more resistant to inducing apoptosis by a lot of things, including viruses.
Right.
So it probably contributes.
Yeah, so I thought that was something we were going to come back to, the age dependence, because the next paper, which I think this was a Beth Levine paper now, there are a series of papers.
She was a postdoc in my lab.
She was a postdoc and then came to Columbia before going somewhere in Texas, UT Southwestern. BCL2 protects mice against fatal alpha virus encephalitis. So what did she do there?
Oh, I think that those were mice.
Same virus, put it in mice.
Same virus, now in mice. But I think that those experiments were using a virus that was expressing BCL2.
And if you put those in newborn mice, which would normally die from wild-type virus, now they don't.
Right.
That's correct, right?
Right. And we had two strains. We had strains that killed adult mice and strains that didn't.
Right.
The persistent virus infection in brains that doesn't kill mice, does it alter their behavior, though?
Oh, so that's a good question. And behavioral studies have not been a strength of my lab. But we did recently I recently had a student who was very interested who was a veterinarian who was very interested in that. And so we looked at mice that were recovering, that were infected with a strain that didn't kill them, and during the recovery period tested them for their memory and that sort of thing. Sure. They don't do as well. So
there you go.
Yeah, right. So there are abnormalities that are associated with this long-term persistence of the RNA in the brain. It's
interesting because if it's just pieces of RNA, it's probably not being translated, but maybe it's inducing something that's causing.
Well, and it certainly is. It inducing, I don't know what it certainly could be inducing something in the cells themselves, but it also is certainly inducing a sort of ongoing immune response in the brain too, which is not necessarily good for your memory.
Exactly. It sounds, I always think of viruses as monkey wrenches in normal cell biology. And these monkey wrenches persist. The cells, even if you cleared the RNA, would the cell be different afterwards?
Oh, I don't know about that, because we never clear the RNA.
Dickson, maybe you have pieces of RNA in your brain.
Well, I was thinking just exactly that, but
and that
I couldn't
could explain
remember whether I did.
Some of the things some of the behavior that you
see. This is Be Nice to Dickson segment.
So here's the story: in young mice, newborns, you infect them with Synbus, and the neurons apoptose, and that's why the mice die. Right? But 4-week-old mice, now they're producing BCL2 and there's no apoptosis. So that's why they survive. That's it.
No, that's not it. I don't think that's it. I mean, I think there's much more to it than that, because you get much more. I mean, I think that was an original kind of hypothesis, and I think it's maybe a contributor. But I think that the fact that the virus can replicate so much more efficiently in an immature neuron than a mature neuron is also a very important thing that I don't think is because of BCL2. I think it's other factors.
Which you don't know. You haven't identified those, right?
No.
Yeah, it's hard.
Do you think SYNBIS could be a tool that people who want to understand brain function could use to make certain things go wrong and then see what that does to the overall pattern of learning? I know they're using other viruses that way too.
Right. And it is tropic for certain types of neurons, entered horn cells, for instance, in the spinal cord, but also hippocampal neurons. So it doesn't infect all neurons the same way. The other thing that gets upregulated in neurons as they mature are various components of the interferon system. So you see higher levels of interferon-stimulated genes, of IRF7, other things that are. So as I say, I think it's probably a multifactorial increase in resistance to virus replication.
But the fact is that young neurons apoptose in response, and that's because they don't have BCL2. That's it.
Well, I don't think that's the sole.
There's something else.
Again, I think there's more to it than that, because, as I say, they replicate the virus. I mean, to 1,000-fold higher.
So I guess my question is, what is the reason that immature neurons undergo apoptosis in response to a stress? Do we understand that at all? And mature neurons do not.
Well, I don't think that we totally understand it, because, I mean, it's the same way if it's a stress of growth factor withdrawal. Also, young immature neurons will die and mature neurons will not. So I think there's a whole programmatic. The host has a lot of investment in having neurons survive. Once they are matured and made all their connections, then I think there's a lot of factors that try to encourage them not to just die willy-nilly when something comes along.
Yeah, I want to get back to that when we talk about immune responses and clearance. But you mentioned the neuroadaptation, so you can adapt SYNBIS to kill older mice. Is that correct?
Right.
And you do that by passage?
Yes, it was just
mouse-to-mouse passage.
Exactly.
And where is the mutation?
The main mutations are in the E2 glycoprotein.
Okay, and I think I remember reading that what that does is it induces apoptosis in adult mice now. Is that correct?
Right. Those viruses replicate more efficiently in mature neurons than the non-adapted.
Despite
virus.
having BCL2 in mature neurons, right?
Right. So you can overcome whatever those inhibitions are if you have a better replicating virus.
Is that what you think it is? It's simply that somehow this E1 mutation allows for better replication that overcomes the inhibitor of apoptosis.
That's our current thinking.
Whether
that's actually the fact is another question.
So since this is in a glycoprotein, is it affecting receptor interactions? Do you know?
Well, the virus certainly infects mature neurons more efficiently than the unadapted virus. But but the difference is not as dramatic as it seems like the phenotype is. So I think there may be more going on, but I can't tell you what it is.
Okay. The other kind of arc of work that I think is really interesting has to do with what you mentioned in the beginning, the idea that antibodies cure a SYNBIS infection. And I think the first paper in 1986, monoclonal antibody cure of lethal SYNBIS encephalitis in mice. And then, of course, in 1991, which I remember when this was published, we all said, what? Antibody-mediated clearance of alpha virus from neurons. So how did you get to that point?
Oh, we basically wanted to figure out, so we knew these mice could survive infection, that the virus was, that it replicated, and then it was cleared within about a week. But if you infected a skid mouse or some mouse that couldn't make an immune response, then the virus, and you had a virus that wasn't going to kill the neurons, then you could have real persistent infection where you could continue to isolate infectious virus over a long period of time. The simple question was when Beth came to the lab as a postdoc was, what aspect of the immune response in this wild-type mouse versus the no immune response in the skid mouse is responsible for that clearance process? So we passively transfer, it's simple experiments that do passive transfer of antibody, and then subsequently mapped what the specificities were. But
So transfer of the antibodies cures the mouse.
To the skid mouse, yeah.
And so these antibodies are getting into the CNS, right?
Right.
Which at that point was shocking, right?
Right.
And do we understand how antibodies get in?
Well, antibodies, actually we did some less prominent type of experiments looking at that. And antibodies get in from the periphery at about 1% of what's in serum. And it turns out you don't need very much. So even after the blood-brain barrier is no longer disrupted by the inflammatory response, you still get antibody in, and it's enough to affect this process.
Now, do you think this is a general phenomenon that for viral CNS infections, the antibodies is resolving infection?
I think it's a very important part of it. Yeah, right.
Because we all think cytotoxic responses are important, but in the CNS that would be crazy, right?
That's exactly right. So that's one of the reasons we were interested in how recovery occurred if it was in a situation where you didn't want to kill the infected cells and get rid of virus that way.
Of course, neurons don't make MHC molecules also,
No.
so they could not be targets of CTLs, right?
Right. That's still a little controversial, but basically they're not. They certainly don't make it easily.
Okay, so there would have to be another mechanism. So one is antibody.
Right.
And if you have a virus infecting a neuron, how does the antibody take care of that? Do we understand?
It binds to the outside of the neuron because the specificity of the antibody is against the E2 glycoprotein.
So, which are displayed on the surface.
Which are displayed on the surface,
Okay.
because this is a virus that buds from the plasma membrane. So, right. But the next step of how that happens has been harder to nail down.
So you think the
There's some
antibody
hints.
binding to the neuronal surface is somehow inhibiting maturation of the particle?
It does do that for sure,
Yeah.
but I don't think that's the sole thing, because we also know it shuts down it shuts down synthesis of viral RNA after a few hours. So it's doing more than just inhibiting budding.
Okay.
Is there any evidence that it gets inside the cell?
We've never, if mostly it stays on the outside. I can't say for sure that none ever gets in, and that the small amount that might get in might be the most important part. But our current thinking is that it's all happening at the surface, and we have put the antibody on beads so that it really couldn't get in, and we still get the same effect
That's a good
of
experiment.
inhibition. So we're pretty convinced it's acting at the surface.
Whatever the mechanism, it has to be non-lytic for the
Right.
neuron, right?
Right.
That's the key.
Right. Yes. Neurons survive this process.
So if you didn't have antibody, could there be other non-lytic mechanisms of curing a viral infection in the brain?
Right. Well, so for our virus infection, antibody is by far and away the most important. We also know, which I'll talk about a little bit tomorrow, that interferon gamma can actually inhibit virus replication and infectious virus, but only in the spinal cord. So it only works for anterior horn cells and hippocampal cells and cells in the brain. Interferon gamma alone is not able to do this. Although I think in reality, the two are acting in concert in a normal infection. Both are contributing, but that's also a non-lytic process.
And so how do you know how the interferon gamma is eliminated? Is it inducing a protein or proteins that somehow?
It's definitely a signaling mechanism that's JAK-STAT dependent. So it's inducing one of the 95 ISGs that it induces is undoubtedly important for inhibiting virus replication.
Within neurons, right? within neurons.
Within neurons, but particularly within motor neurons. So anterior horn cells, motor neurons, actually motor neurons of the brainstem as well. But so that's intrinsically interesting too, and we don't know. All neurons basically express the interferon gamma receptor, so it's not just a receptor thing. So it must be something about the signaling pathways that are present in motor neurons that aren't necessarily present in other kinds of mature neurons.
So having the ability to knock out genes has clearly impacted this field a lot. Like not just interferon receptors, but other things. How about the ability to make neurons from, induced stem cells, organoid cultures? Have they contributed now, or will they?
Right. Well, they certainly could, and those are the kinds of things we're looking into, trying to do more of that sort of thing. We really haven't perfected culturing motor neurons from the lumbar spinal cord, but those technologies are now more widely available.
Yeah,
so we're tackling them.
I could imagine that would make it a lot easier to sort out some of these questions.
Exactly.
So some of the other things I noticed I wanted to touch on, you had a couple of papers on strain differences in susceptibility in mice, that is. And I'm wondering if you took that any further to identify loci that might be involved in susceptibility.
Right. So I had one PhD student who was very interested in that and got as far as identifying a QTL on chromosome 2 that was, and it was a difference between BALB/C mice and Black 6 mice, which are pretty dramatically different mice, actually. But so it was mapped to a QTL at the time that student was working. He didn't have, the mouse genome was not sequenced. So it's a project we should really go back to, because now the tools are much better for having a chance of. But again, I think it's probably a multigenic trait. That was one important factor, but that certainly wasn't the only genetic factor between those two mice.
I noticed you had a paper where I think you did retroviral disruptions, and you had a cell line with a single disruption, and the virus didn't bind to that anymore.
Right.
Correct. And what was the gene involved?
It turned out it was an enzyme involved in the heparin sulfate pathway, and we know that heparin sulfate is a very important binding factor for this this virus, and so it turned out to be an answer we already knew, basically, by identifying heparin sulfate as a.
Is there also a proteinaceous receptor for the virus?
Well, a number have been identified by us and by other people. I don't know that any. The most recent was NRAMP. So I'm not sure that any are really totally accepted or explain the tropisms or the mosquito versus. I mean, avian, mosquito. The receptors for alpha viruses have to be either very broadly expressed.
Ones,
like for all the arboviruses, actually, or they have to use lots of receptors. My personal opinion, they probably use lots of receptors. Lots of different things can serve as receptors.
Okay. And there is an individual at Johns Hopkins who is very interested in sex differences that impinge on virus replication, Sabra Klein.
Right, yeah.
So I was wondering if you've ever looked at that for SIMBIS.
We have, but I can't say we've looked at it rigorously. In some situations, we do see some differences, and mostly we have looked at both sexes and haven't appreciated that there have been differences. So.
Yeah, she just talked at ASV, and so many interesting aspects of that.
Yeah.
And now the recent paper showed there's a Y chromosome locus that influences influenza virus susceptibility. So I'll bet we're going to see more of that for other viruses as well. Among the other things you've worked on measles and many different topics, atypical measles, immunosuppression, encephalitis. So at about 1 in 1,000, measles gets into the brain. Is that correct?
The incidence of, I don't know how often it gets into the brain. That's not been an easy thing to answer, but about 1 in 1,000 get SSPE, especially if they were infected at a young age. So subacute sclerosing and panencephalitis is a neurologic disease. It's a late consequence of measles. It's due to measles infection, but so we know it gets in there. But now we also know that measles persists in the periphery for months, maybe years in lymphoid tissue. And there was just a recent paper saying that, well, it's actually 1 in 500 if you're under 2 at the time. So I think it's age dependent how likely you are to get an infection of the brain, and whether it's common and most people recover, or I think that those are the kinds of things we don't know. CNS abnormalities are common in measles, but like cells in the cerebral spinal fluid and that sort of thing is.
How does the virus get into the CNS? Do we know?
I don't have any idea.
Maybe would it be lymphocytes or?
It could be. Yeah, that's how it gets in most places, because it infects both B and T, and as well lymphocytes as well as monocytes. So that's certainly how it circulates and how it infects many tissues during systemic infection.
You've also done a lot of work on measles vaccines. And I'm wondering, WHO is targeting that for elimination after polio. What do you think about that?
I think it's a
Wait a minute,
great idea.
eliminating the vaccine or the disease?
No, the disease.
Just be clear about that.
We have a good vaccine. So, interestingly, we don't know how the vaccine works. I mean, we don't know what attenuation is. It's a live virus vaccine. We don't understand attenuation for that vaccine.
Does it persist a lot in the tissues?
It's been very little studied. We understand very little about the measles virus vaccine, actually, because once it worked, that was sort of the end of what people were interested in looking at.
That wasn't the case with polio, because after polio vaccines, people wanted to know how they worked, and that was really sorted out pretty well. Identifying the attenuation determinants.
Right. Yeah, that's not
measles.
happened with measles.
Is that because there's not a good animal model, or?
No, there's a good animal model, because monkeys are a good model. Yeah.
Yeah, the same thing with polio, so I don't understand. Anyway, getting back to eradication or elimination.
Yes.
WHO says we don't call it eradication.
No.
We call it
We call it elimination, right?
So
elimination has happened for measles in regions, but eradication is going to be hard.
We're almost there with polio, but the hard part is not the science part, it's the political part.
Oh, it'd be the same way with measles. If you gave the vaccine enough and to enough people and effectively enough, then you could accomplish it. But we have a lot of trouble with that.
I always wondered why you never worked on polio, because it's a lovely model for CNS infection.
You were working on polio.
No.
We don't need more people working on polio.
No, I don't believe that. I asked Mike Bishop once, why did you stop working on polio? And he said, because you started, and I just don't believe it. I'm sorry. It was never...
Well, they also went through, the NIH went through a phase of discouraging people from working on
That's
measles.
True.
They certainly were discouraging people from working on measles, but they also discouraged people from working on polio because it was thought to be not a problem.
I suspect for you, because from the start, SINBIS had a mouse model.
Right.
Whereas for polio, we had to make one. It wasn't
That's
until
right.
the
Well,
late 80s, right?
Right, yeah.
So that
exactly.
probably, by then you were ensconced in SINBIS, so we weren't going to work on another
I
virus.
wasn't looking around for another virus.
What's early in your career, you worked on Visna virus.
Right.
How many people here know what Visna virus is? A few.
Some people.
Don't have a clue.
Right.
Few. Why did you do that? What is the Visna virus?
So Visna virus is a lentivirus. It was one of the first viruses that, and HIV is a lentivirus, but this was worked on long before HIV. It's a disease of sheep, and it was a, quote, slow virus disease. So you knew that you could infect sheep, but sheep didn't get sick for a year or two after they got infected. And so this was in Dick Johnson's lab, and as I say, everybody had all the fellows each had their own virus. Well, Bill Narayan had decided to work on Visna at that time. He was a veterinarian, and he was interested in sheep as well. But the other thing, the way the lab worked was that people also were contributing various aspects of expertise. And so I was sort of considered the immunologist. So if you had an immunologic question or an immunologic component to your virus, then I often got involved. So I got involved with a lot of the early immunology with Visna virus infection in sheep.
What I think is interesting, this is pre-HIV.
Right, I know, it was before HIV.
Lentivirus, and a lot of people were interested because of the long development of, as you say, of disease and so forth. And this is a sheep disease,
It's
right?
a sheep disease.
And how is it spread? Contact between sheep or respiratory?
Yeah, that's a good question. I imagine probably.
So then you went on to work on HIV, and I always thought that presaged your work on HIV. It's quite interesting.
Yeah, well, it was. That's how we, because that early work on Visna was some of the only pathogenesis or kind of virology work that had been done on lentiviruses. When they all of a sudden discovered that HIV was a lentivirus, then yes.
Yeah, so then we could go back to all the other work on other lentiviruses and learn quickly.
Right.
This idea of slow, you made air quotes when you said slow.
Well, yeah, well, there were a group of diseases that were called slow virus diseases, and that was one of them.
We don't use that term anymore.
No, because
right?
now we know a little bit more.
I have an old book on my shelf. It's Slow Virus Infections, it's called. It's chapter after chapter of different examples. And there was a Swedish scientist who
Right. Icelandic.
Sigurdsson,
right? Yeah. Who formulated that concept of slow virus diseases.
So more recently, you've published a few papers on Zika virus. What got you interested in that?
I was just intrinsically fascinating virus.
So is it the neurovirology aspect, do you think?
It was. And also, actually, what's really been fascinating to me is the Guillain-Barré and trying to understand what that, because we don't, so Guillain-Barré occurs sporadically a lot, but almost never in association with and it's considered a post-infectious neurologic disorder, which it is, but mostly the infection and the link to the infection to Guillain-Barré is not known. How those, Campylobacter is one of the few examples where we actually understand what the actual mechanism is of infection followed by Guillain-Barré. So to me it seemed like, and I think that's still true, because I don't think a lot of people are actually looking at that aspect of the disease. It seems like a good opportunity to have another virus or another infectious disease where you could possibly understand what the link is and whether, is it really a cross-reaction between the virus and Schwann cells or, exactly what's going on.
So there's never been an animal model for Guillain-Barré, right?
Well, there's experimental autoimmune neuritis, but that's you inject, myelin, peripheral myelin. It's like EAE. You inject the myelin, and the animals get a neurologic disease.
The old rabies vaccine.
But there's no model where you can actually, other than the Campylobacter, where you can actually use an infection and then get a Guillain-Barré in an animal.
Okay. All right, so let's wrap up. We have about 10 minutes left. Got some philosophical questions for you.
Yeah, I'm not so good at those.
Come on, you have a PhD.
Right. Yeah, right.
So you worked a lot on viruses that get into the CNS, and I've been thinking about this a lot lately. In my view, the CNS seems like a viral dead end because once you get in, the virus cannot leave. So
That's right.
You agree with that?
I agree with that.
Okay. So then there's really no selection for a virus to be able to infect the CNS directly because it's a dead end. And any virus that got in and replicated well would never persist in the evolutionary sense because they can't get out.
The transmission, it wouldn't be clear how you'd transmit it, right?
So, but obviously many viruses can get in and replicate. Right. So it must be a property that's related to something else that happens. So you replicate well in a blood cell, and maybe that lets you replicate in the CNS. Does that make sense to you?
Yeah, I think for most CNS infections, so no matter what kind of infection you're talking about, they're uncommon complications usually of common infections. So I agree that from a teleological point of view for the virus, it's not necessarily smart strategy.
The reason I mention it, particularly lately with Zika, the popular press has picked up what scientists say, and they're saying, well, the virus evolved the ability to replicate in the CNS, and it just doesn't make any sense, right?
Yeah, no, I agree.
It may be a consequence of something else, as we have said. Okay, so over 300 papers. Which one would you say is your key experiment? Only one.
Only one.
And nobody in this audience is on any of them, so you don't have to worry about insulting.
That's right, insulting anybody. Well, probably the discovery that antibody can inhibit virus replication intracellularly in a non-cytolytic fashion.
Yeah, that's pretty cool. I would agree with that. I would like to have done that one too. Okay, of everything you've done, this is a different question. It sounds similar. Of everything you've done, what do you think is your most important contribution to the field of science?
Oh. I usually think those are the people you train rather than the actual experiments that you do.
Very good. I ask everyone because I want to hear.
Right.
Not everyone says the people, but I'm glad you did. You had a question, Dickson, right?
No, I would love to hear your take on today's climate for funding research, because there's a lot of discouragement out there, as we all know, with regards to the future of this country over the next, say, 4 to 8 years. And people are now trying to decide what to do for their livelihood. And do they want to go into science or don't they? And then they hear all these discouragements about it's so difficult to get grants nowadays, and then you have to establish your lab, and there are not enough jobs, and there's an anti-intellectual climate in this country. What do you think about all that stuff?
Oh, I think all of that's true. But I think that you have to encourage people to, you don't know that what it's like now is going to be what it's like 5 years from now when you graduate, or after you finished your postdoc, or whatever, that things change, and that you have to do what you really want to do, and you're usually better at doing something you really like to do, and if that's science, that you should pursue it as long as you can and as hard as you can. But I agree that trainees right now are very, well, they're just worried, that they won't be able to, quotes, make it.
That's right.
But your advice is if you're passionate, do it.
Do it, right.
Hear, hear.
It's a good title.
That's right.
If you're passionate, do it. Right. If you had not become a scientist, what would you have done?
Gosh, I don't know. That's a good question. Yeah, I don't know. Pretty much everything I really like doing relates to science in one way or the other.
That's an answer. That's what I would say. But we get really interesting answers there. If you could have dinner with anybody, living or dead, who would it be?
Gosh, I like having dinner with a lot of people.
Somebody who's not around anymore that you might have been interested in.
Yeah, right. I don't know, it'd probably be, I don't know, there's, if it was somebody who wasn't around anymore, some of the maybe early pioneers in thinking about viral pathogenesis, trying to think of that. Now I remember
early
the name.
Pioneers in viral.
Louis?
Well, no, Louis, I don't know if Louis would be so interested.
I don't either.
How about Tom Rivers? How about that?
Yeah, somebody like that. And also, there was the Australian.
Frank Burnett?
Yeah, Frank Burnett. I mean, some of the people like that, yeah.
Mm-hmm. Well, Frank Burnett was around until recently. You never met him?
Never met him, yeah.
But Louis would be great to have dinner with just for the wine. Because he's
right.
wine industry.
I think he knew his way around.
Yeah, I think he did, actually.
I think it would be really
cool.
Einstein,
he'd be interesting too, but I'm not sure I could keep up my end of the conversation.
We could talk to him about viruses, because he would not have known anything.
Oh, okay, all right.
Anyway, and finally, last question. What's in the future for you?
Oh, well, that's a good question. So for right now, I don't know the answer. Right now, I'm still running my lab, and I'm still teaching, but I'm no longer chair of my department, so that's good. But I'm also vice president of the National Academy of Sciences, which is a half-time job. So I could probably keep doing it. I'm doing that for at least another 4 years, so good. We'll see.
Sounds good.
So for right now, no plans to change.
Great. And you still have a lab that keeps publishing and doing science. Yeah. And you'd like to do that as long as you can.
I love it, yeah.
Okay,
right.
Very good.
Nothing better than a new piece of data you didn't expect.
Absolutely, totally agree. All right, this is This Week in Virology, TWiV. You can find it at Apple Podcasts. You can find it at microbe.tv/TWiV. If you have a cell phone or a tablet, any mobile device, there are applications that play podcasts. If you haven't done it already, just get one of them, search for TWiV, and subscribe. It really helps us if you subscribe, even if you don't want to listen. I don't
Really got me out here.
don't know why you wouldn't want to listen, but subscribe, and that helps us because we need financial support. Dickson is here actually because of our fans. They have
true.
contributed funds, and you can go over to microbe.tv/contribute. We have different ways you can support us. We have a Patreon account where you could give us $1 a month if you'd like, or other ways as well. We do have ads, as you heard up on top on the show, but our main support is from our fans. And we'd like to do more and travel more and do podcasts to expose the world to science. So if you like that, help us out. We also love getting questions and comments from our listeners. We typically make them part of the show. We didn't have enough time today for that, but you can send them to TWiV@microbe.tv. I want to thank Diane Griffin, Johns Hopkins University, for joining me today. Thank you so much.
Thank you for the invitation.
Great hearing about your career. Dickson de Palmier, you can find him if you want to see a really cool fishing website, thelivingriver.org. I helped him build it, but it's got all his life experiences, photographs.
Ecology of the trout stream.
You got to use the mic, Dickson.
It's called the ecology of the trout stream.
It's beautiful, but if you want parasitology, what is it? Parasiteswithoutborders.com, right?
Exactly right, exactly right.
Dickson.
Little ones and big ones.
Thank you, Dickson.
You're welcome, Vincent.
I've been working with Dickson since the beginning of all of these shows, and I really appreciate it.
Yeah, well,
even though I give you a hard time. Thank you very much. I want to thank the organizers of this meeting for having TWiV here. My contacts have been Marshall Bloom and Ted Pearson, so thank you very much. And Toby Boland for doing the behind-the-scenes stuff. I appreciate that very much. I want to thank the sponsor of this episode, Blue Apron. Go check them out at blueapron.com/TWiV. The music you hear at the beginning of TWiV is composed and performed by Ronald Jenkins. He lets us use his music. You can find more of his work at ronaldjenkins.com. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.